morning, dear students. Let us begin the class with a small prayer. Lord Jesus, we surrender all medical professionals into your heart. Bless us with your love and care to provide healing hands and smiling face to all sick and suffering. Amen. Ave Maria. In the last class, we completed the structure of cell in detail on nucleus because our concentration is on gene. Gene is situated in DNA. So we have learned the structure, microscopic structure of DNA. Here in this class, we are going to study the major function of gene or DNA. Our body is made up of proteins. There are different types of proteins in our body, and the proteins are divided as amino acids as a process of digestion. Whatever the food that we have taken, the protein content of our food is digested and it is broken down into amino acids. And the small amino acids are present in our body. And how it is from the amino acids, different types of proteins. As you know that our body is constitute of different types of proteins. Some act as bodybuilders, but some forms hormones, some forms antibodies, some forms enzymes. Yes, totally when we look into the small, minute 20 amino acids forms this entirely different human body. And it functions in many ways even till the death of the person. So how this protein is synthesized in the body?
And absorb. And where
Protein is synthesized mainly through the process called gene expression. Gene expression. Gene is expressed. Okay, when we express ourselves, when we want to demonstrate something, okay, here the gene is expressing so that one part of the DNA is duplicated and forming RNA. And this RNA will act as a template to form proteins. Okay, we are moving to study that. So, in the process of gene expression, okay, it constitute two processes, that is transcription and translation. Transcription and translation. What is transcription? Okay, forming messenger RNA from DNA is known as transcription. Transcription is happening within the nucleus. The messenger RNA which is formed during transcription is translated into protein through translation. So, the gene expression contains two processes, that is transcription and translation. Now let us see the transcription in detail. Okay, before moving into the transcription, as you have seen in the video, the DNA and RNA, it contains sets of nucleotides That means a 3 base pairs maybe, okay, so adenine, guanine, cytosine, okay, like base pairs are continuously found in DNA. Within that, okay, few members forms a full family, isn't it? The same way, 3 base pairs forms codons. This codon is acting as a unit, and this unit specifies the amino acids. So everywhere, codons, 3 nucleotides. Okay, so in the process of transcription, mRNA is forming from DNA. mRNA is forming from DNA. And in every process, we can see that there is an initiation stage, elongation stage, and termination stage. Okay, at the initiation stage, you have seen in the video, an RNA polymerase is coming and getting attached to a certain portion of a DNA because that portion is known as promoter gene. It is known as promoter gene or an initiator. Okay, in this, one codon is acting as an initiator, and this RNA polymerase get charged when it come towards this particular codon which act as a initiator. It may be UCC or it may be AGG, and it act as an initiator and it gets attached to this particular DNA. Once the RNA polymerase get attached to this DNA, we can see there is an unzipping, zipping and unzipping. Okay, we are putting the zip of a dress. So what do you mean by unzipping? Okay, just opposite to it, we are removing it. So it is otherwise called as unwinding, just separating the two DNA. Okay, and now the DNA, as you see in the picture, it has got a two areas, and one is complementary to each other. If it is above thymine, it is cytosine. Clear? Okay, guanine, cytosine, thymine, adenosine. So we can see that is a complement each other. So now one pair of it act as, okay, one of the pair act as a template. So based upon that, what will be forming? Messenger RNA will be forming. While forming the messenger RNA, uracil will be formed. That is only difference between the DNA and RNA. Clear? Instead of thymine, uracil will be forming in RNA. So now just if it is adenosine, uracil will be forming. Okay, guanine, cytosine will be forming. If it is down, it is guanine, above cytosine is forming. Thymine means adenosine is forming. So just complement to each other, it is been forming. Okay, so now the continues. Okay, so RNA polymerase continues this process, controls this process till it reaches to another point where this particular codon act as a terminator. After that, it will not move on. Okay, then with that the process discontinues and back the DNA is coming back as a normal stage. Okay, it will come back to the same shape. Okay, now what formed? One messenger RNA is formed. Hope you understood what I have explained. The initial event is unzipping of DNA and creating a single strand of messenger RNA. The enzyme RNA polymerase unzips the region of DNA, and the DNA serve as a one side of the DNA serve as a template for the creation of messenger RNA. The messenger RNA is mirror image of a DNA nucleotide sequence. Mirror image only with the exception that uracil is formed instead of thymine. After RNA is transcribed, the DNA is rezipped and available for the future use, and even RNA polymerase is getting disconnected from the DNA. Now in our hand, we have a smart, small messenger RNA. Okay. When children are forming, they have many behaviors which are very good, and there are many behaviors which are not good. So what happens? The parents will control the behavior which is not good, and they will enhance the behavior which is useful. So when the person gets mature, if he is nurtured well, the unwanted behaviors are removed and the good behaviors are encouraged. Yes, it is happening naturally in every creation. The same way, the messenger RNA which is formed now is immature. It has got both useful part and unuseful part. Okay, the segment of mRNA that codes for a protein is called as exons. Useful part is known as exons. Clear? But in between the exons, there are some other part, that is non-coding sequence, that is called as introns. Okay, so exons are there, it is good, it is essential, but it in between, introns also are present in the messenger RNA. We don't want these introns. So what to do? Yes, introns must be removed. Yes, there is a process happening after the formation of messenger RNA. Okay, the introns are removed. Introns are removed. Now, when you look into the picture, you can see this yellow part is exon and this red part is intron. So, this yellow is good and we need it, but intron we do not want. So, what to do? It has to be removed. It has to be spliced. It has to be spliced. So, there is a hormonal action comes, it comes together, and what happened? The spliceosome, okay, and it cut down the introns, just like how the parents are cutting down the unwanted behavior of a children and forming a mature person in future. This various hormones which comes and act in the introns, and it cuts these introns and unites these exons together. So after once the splicing has completed, now RNA is free from introns. That means it is a mature RNA only with useful part, the parts which are coding for protein. Yes, this mature RNA is formed, and this mature RNA has an ability to pass through the from the nucleus, through the nucleospore. It passes from the nucleus to the cytoplasm of a cell. So transcription is getting completed within the nucleus. So the next stage is happening within the cytoplasm. Hope you understood what is splicing. It is the removing of unwanted introns, which is a non-coding sequence, cutting out and forming the exons, the coding or even the segments that code for protein. These exons are brought together and the introns are thrown out from this messenger RNA. Now it is ready for, yes, now it is ready to form the particular protein for which the gene has started to function. Now we'll see the second step of protein synthesis, that is translation. What is commonly translation? Now I am speaking to the language which you cannot understand. Somebody else will translate into the term or even into the language which you can understand. So that is called as translation, okay, in commonly. So what is translation in protein synthesis? Yes, now we have the duplicate of a DNA or messenger RNA. This messenger RNA must be converted to the language of amino acids to form protein. That is known as translation. Okay, translation is mainly happening in cytoplasm with the help of ribosome. Okay, the ribosome is involving in the formulation of or in the process of translation. Clear? So the messenger RNA, which has come from the nucleus, which has mRNA, and based upon this mRNA codon, now another RNA is being formed. It is known as tRNA or transfer RNA. It is known as tRNA or transfer RNA. Okay. If you want, you can just move on to the next slide and you can see that these are the number of amino acids that we have in the body and the corresponding codons. If it is GCA, it is alanine. Okay, and along with GCA, GCC, GCG, GCT, guanine, cytosine, adenosine, that is GCA. Guanine, cytosine, cytosine, GCC. Guanine, cytosine, guanine, okay, guanine, cytosine, thymine. All these act as a alanine. So we can see that there are, okay, more than one codons that indicates one amino acid. Okay, so this DNA template which is present, okay, in the messenger RNA, that forms the corresponding transfer RNA. The process of translation can be said as 6 steps. Okay, in the first step, what happened? The messenger RNA is binding to a small ribosomal subunit. Okay, within the mRNA, AUG, the codon AUG is an start codon. So once the AUG comes, okay, where there is AUG in a messenger RNA, there the small portion, the small ribosomal subunits gets attached to it. So we can see that with that, the beginning of translation happens. And opposite to it, complementary to it, okay, AUG, the transfer RNA UAC is getting attached to the messenger RNA and it has an amino acid in it. Clear? So This AUG is a codon for the amino acid methionine. Thus, methionine is always the first amino acid in a growing polypeptide. So, UAC is a start codon, and even there are few stop codon. Once it reaches to the start codon, the process of translation begins, and once it reaches to, there are three stop codons, any of the stop codons, it completes the process of translation. So in the first part, the ribosomal subunit is getting attached to the start codon AUG of messenger RNA, and corresponding tRNA UAC is getting attached to the mRNA with the amino acid methionine in it. In the second step, the large ribosomal subunit is getting attached to the small ribosomal subunit. Now the down we have small and the above we have the large ribosomal unit. To the mRNA, small ribosomal unit and the large ribosomal unit. This is the functional ribosome. The initiated tRNA with methionine fit into the P site of ribosome. We can see that in the large subunit, there are three areas: E areas, P areas, and A areas. And the first tRNA for the methionine is fit into the P site of the ribosome. Okay, that is the second one. The third one is the anticodon of another tRNA is getting attached to the second portion next to it. Okay, so that is getting stimulated, and the corresponding tRNA is coming with its amino acid, and it is getting attached to the S site of the ribosome. Okay, so the second tRNA is coming and getting attached to the A site of a large ribosomal subunit. Okay, so that is the third step. Now the fourth step, the large ribosomal subunit forms a peptide bond between the methionine and the second tRNA, which is at the A site. Okay, that means the tRNA which is present in the P site and A site, and these both carries amino acids. There is a peptide bond is formed between these two amino acids. Okay, and that is the fourth stage. In the fourth one, there is a peptide bond is formed between the amino acids that is present at the tRNA of P site and the S site. Now the fifth step. After the peptide bond formation, what happens? The amino acids get attached to the S site tRNA or even the S site amino acids. This particular RNA leave the P site and the tRNA from the A site move to the P. And how it happens? It happens after the ribosomal subunit is moved towards the DNA. So this template, okay, or even the codon of P site is moving to the E site and it is getting removed from that. The codon which was present in the S site or the transfer RNA which was there in the S site moves on to the P site, and another tRNA is coming complement to this next codon is coming and getting attached to the S site, and again what happens? A polypeptide bond occurs between the amino acids and it is getting attached. Amino acids are getting attached. So 3 forms, like 4 forms, 5 amino acids are brought, and how many numbers or amino acids are required, that many numbers are brought. And once when this ribosomal subunit reaches to any of the stop codon, okay, with that it is getting completed. So that is the sixth stage. Okay, so when it is getting attached to the any of the stop codon, with that process, with that the process of amino acid or the amino acids is brought and the protein molecule is being formed. And now we can see that it is not a completely finalized protein, and it is just like I call it is a small protein. This protein will have to undergo and changes in endothelial cells, and finally it forms a mature protein. Look at the picture. We will see the translation again. Okay, the translation begins when the small ribosomal subunits get attached to the initiated tRNA, that is AUG, and the transfer RNA and the corresponding amino acids is getting attached to the P site of L. Okay, so that is the first one. And the second one, what happened? This large subunit of the ribosomal unit is getting attached, and the tRNA is placed in the P site of a large subunit with its amino acid. The third one, we can see that and another tRNA is coming into the A site, and the fourth stage polypeptide bond is formed between the amino acids. And in the fifth one, we can see that the first initiator codon is getting removed from the P site, and the second one is moved into the P, and another one is coming and getting attached to the A site. So in the same way, repeatedly the different types, okay, the different colorful, okay, different color it has shown, that sees that the different amino acids are coming, it is getting attached. So a large, these amino acids are brought together to form a large protein molecule, and with the stop codon it gets completed. So I am concluding the protein synthesis. Okay, protein synthesis is happening mainly through the gene expression. Gene expression has got two processes, that is transcription and translation. Transcription is happening in the nucleus and the translation is happening in the cytoplasm. The transcription where the messenger RNA is forming, where the DNA is acting as a template, and the messenger RNA which form, it has got both useful and unuseful part. It is known as exons are useful part and the introns are unuseful part. Through the intron splicing, a mature messenger RNA is formed, and through the nucleosome it is transferred from the nucleus to the cytoplasm, and in the cytoplasm translation begins with the help of a ribosome. Okay, the small subunit is getting attached to the start codon, and the tRNA corresponding to the messenger RNA is getting attached to it, and the larger ribosomal subunit is getting attached, and each time two transfer RNAs are coming to the P site and the A site, and which is coming to the A site, okay, the amino acids which is brought, it is getting attached to the amino acid which is present in the P site, and so it occurs in different steps, and different amino acids are brought according to the need of a body's protein, and even by bringing different amino acids, the required protein is synthesized in the body. This is how the proteins which act as a different steps. Okay, protein is a body building blocks, and everything, every aspect of our body is constituted of proteins. Yes, each protein is synthesized in this manner. So I hope you understood how the DNA will act, or how the DNA will control the total bodily functions, mainly through the protein synthesis. Hope you understood the class. If there is any doubt, please be free to ask me. I will explain to you again, and watch the video clearly 2-3 times even after listening to the explanation. and read the note. And still, if you are not able to follow, when you are coming on Saturday, please ask. I will explain it. Thank you.